Hey, Mitchell Harmatz here. So, what are you? Well, you're you, of course. But how would we define you scientifically? The closest that we've come so far is DNA. But you're more than that. You are the sum of your experiences. You are what you've been through. But how can we display this? How can we represent this? What you are? This representation is exactly what Sebastian Seung is working on. He runs his own lab at MIT and is working on something he calls a connectome. A connectome is a 3D model of the brain. Think of it like a roadmap showing every single connection in the brain. A brain is made up of neurons, long tree-shaped cells that branch out at the ends into things called dendrites. And each of these dendrites are connected to other dendrites. This connection is called a synapse. These synapses are what is really important. These connections are what a connectome attempts to map out. They represent both the sum of your experiences and your DNA, both nature and nurture. Certain connections strengthen, new connections form, and some connections fade away and can disappear depending on what you experience. Dr. Seung compares it to a river. Your DNA is like the riverbed, providing the initial shape for the river, and your experiences are like the water in the river. The water changes the river as it flows, shaping where it goes. Thus, your experiences shape who you are. Now, why is this important? Why should we study this? Why should we know this, besides the fact that it's pretty darn cool? If we could map out a connectome, a base human connectome, like what was done with the Human Genome Project, with a base human genome, then we could take the connectome of people suffering from neurological disorders and look for abnormalities, places where there is miswiring, so to speak. In people suffering from things like seizures, autism, ADHD, we could look for which neurons are firing too much and which ones too little, where there might be damage, and we could thus identify the source of problems and pinpoint these spots for treatment to help these people with these disorders. We would see, during a seizure, these specific neurons that should not be firing are really active right now. Maybe then using an electrical implant, which hundreds of thousands of people currently use, we could then target this specific area right there and stop the seizure with the press of a button. So, how far along are we? How much have we gotten done? Well, I'm happy to tell you we have a complete connectome of C. elegans. This worm is C. elegans. It has around 300 neurons with 7,000 connections. These sound like pretty big numbers. We did a good job. Well, the human brain is suspected to have over 100 billion neurons, with who even knows how many connections. So we've got a little bit more work to go. In Dr. Seung's lab, they divide 3D images of the brain at a cellular level into cubes, and they color each neuron in a different color. Then they do this for the cube next to it, keeping each neuron the same color, and then another cube, and then the next cube, and then the next cube, and then the next cube, until they've traced out a complete neuron with all of its connections. Now this takes a lot of cubes, which is a lot of work, but it's being crowdsourced currently. There are websites where anybody can go on and color in cubes, and thus millions can help out at once. I personally think it's going to go the route of the Human Genome Project, where somebody somewhere will have an amazing idea which will revolutionize how we trace out the cubes, and it will be done much faster. Next thing that I want to talk to you about is a certain field of neuroscience called optogenetics. Optogenetics was co-founded by this man, Ed Boyden, who is doing some really amazing work. It's essentially controlling the brain using light, causing synapses to fire or not fire using different frequencies of light, different colors. There exists a certain surface protein called channelorhodopsin. Some types of algae have it. When a particular frequency of light hits it, in its case frequencies corresponding to blue light, it allows for the passage of sodium ions, causing an electrical impulse in a cell. The theory goes that Ed Boyden would locate the section of this algae's DNA that codes for channelorhodopsin, extract it, replicate it, put it in a modified virus programmed to target specific cells in the brain of a mouse, mice before humans, of course. This virus would then fly off, land on these cells, inject the DNA in. The new DNA would then be incorporated into the DNA of those cells, and lo and behold, these cells would grow these proteins. This sounds really complicated. Well, because it is. So, how much have we done? How many steps have we gone through? Well, we've actually done it, and it's worked perfectly. These cells have grown these proteins. These cells can now be activated using light. That's absolutely incredible. One type of cell specifically, which Dr. Boyden has targeted, is basket cell. Basket cells are sort of like an off switch for a neuron. When they are activated, they do not allow for the neurons they are attached to activate themselves. One practical application of this is with seizures. When a person is seizing, certain neurons are firing too much, and they don't stop firing. And these basket cells, which would normally stop this overactivity, are atrophied. They're damaged. Now, if you could activate these basket cells manually using light during a seizure, the seizure would stop. These basket cells would cause these neurons to stop firing, and the person would be fine. Another practical application of this technology is with PTSD. Dr. Boyden exposed mice to a small shock, nothing very painful, just sort of annoying, whenever a tone was played. So they associated this tone with the shock and would freeze up and act scared whenever the tone was played, even when there was no accompanying shock. This stimuli caused a scared reaction, even though nothing bad was happening in the later tones. Dr. Boyden then caused the neurons responsible for dopamine to grow the proteins. Thus, whenever light would shine, dopamine would be released. Dopamine is sort of like the feel-good chemical of the brain. You get it as a reward when you do something good. Dr. Boyden then played the tone again for these mice, but now caused dopamine to be reduced, so a good feeling would now become associated with the stimuli. And sure enough, pretty soon these mice stopped having a scared reaction whenever they heard the tone. They just continued on as normal. The mice without this, the control, they continued to act scared. So this application really does work. Obviously, it will have to be changed for use on humans and PTSD, but the application of stimulating dopamine receptors can really be expanded upon. There's a form of blindness in which the photoreceptors on the eyes are damaged. A simplified manner of describing how vision works is photoreceptor proteins on the eyes pick up stimuli, the light coming in, and they then pass this data back all the way to the visual cortex, where it is processed and deciphered into what we actually see and register. Dr. Boyden took mice with damaged photoreceptors and grew chanellorodopsins in place of the photoreceptors. And these mice could now detect light. They could essentially see. He tested this method by putting mice in a 6-armed test chamber with a little water in the bottom, just enough so that the mice could not sit still. Only one of the arms was dry, and this arm had a light shining in it. The blind mice, the control, would pick and choose randomly until they got to the right chamber. But the mice which had their photoreceptors replaced would immediately go to the dry, lit up arm. These mice could see. It worked, and that's absolutely mind-blowing. Now let's get into a little idea of my own. What if Sebastian Seung is successful? What if he makes a perfect connectome? We could then create a 3D model of a person's brain using a computer. And say, using this model, we could identify which neurons were firing at which moment in what order, and record this as a set of ones and zeros, 1 for firing, 0 for not firing. Neurons firing is how we experience something. They process the stimuli and fire accordingly. But what we ourselves register is the firing. But if we turn the order of firing into a series of ones and zeros, have we not just turned an experience into binary? Because that's what the experience is. It's those neurons firing. That's sort of scary, turning an experience into a code. Now to take it even further, What if we combine this with Dr. Boyden's research? We could put different proteins that respond to different frequencies of light on all of the neurons, and then we could shine specific frequencies of light in specific areas of the brain in the exact same order as the binary code of a pre-recorded experience. And if we've done that, have we not just transmitted an experience? Could we not cause someone to experience something without it actually happening to them? Because to them, their neurons firing is all that they register, and their neurons will be firing in the exact same way as if they were actually having that experience. This sounds like mind control, something out of a sci-fi film. Well, because it is. But this research could really lead to something like that. This is incredible, but it's also really scary. It could be used in so many good ways, but could also be used in so many bad ways. For example, if someone is suffering from a condition which causes memory loss, like Alzheimer's or amnesia, and they're losing their experiences, we could simply have them experience things again by causing the neurons to fire in the exact same order, thus regaining that memory. But what if someone wants someone to experience something that never actually happened to them? I don't want that. No, my memories are my memories. I don't want to think I experienced things I actually didn't. Now, is this actually going to happen? Probably not, because a big thing in science is ethics. Is something ethical? Should we be doing it? In order to get funding, research often has to be approved, and that's where ethics comes into play. Additionally, we'd need things like supercomputers to perfectly process the firing in the exact order, and a perfect light source to fire in the exact area. But these are things that could come in the future. Another possible flaw in this is that each brain is different. Each connectome is different with different synapses, though there is probably a base human connectome, but there will be variants. It's just an idea, just a concept, but it can really make you think. Now the real question is, how do we know that this hasn't already happened? Is there a way to know? Am I just hooked up to some machine somewhere? I don't know. This research is saying that it might be possible. Who knows? I leave you with a quote from the philosopher Descartes. I think, therefore I am. You are. But what are you? Thanks for watching.